this lecture series on biochemical engineering, and it's titled receptor-ligand binding. Receptor-ligand binding is something that's ubiquitous in physiological engineering, so to say, and a large fraction of the biological processes or biochemical processes that occur in a human body or any kind of physiological systems are resultant from receptor-ligand binding. So, what is this process of receptor-ligand binding? That is one of the things that we will study, and then we will study with respect to one particular disease, which is a well-known disease, and it's kind of very prevalent in India. So, and with that we will just use the disease as an example to study the receptor-ligand binding, but initially we will try and understand the mechanism of receptor-ligand binding, the kinetics of receptor-ligand binding, and how the kinetics affects certain issues with receptor-ligand binding. Then we will look at a physiological aspect or a pathophysiological aspect of it, that is some of the diseases. So, as I said that many of the processes most of the biochemical processes rather are result from receptor-ligand binding, or in other words receptor-ligand binding plays an important and in most cases a limiting role in these biochemical pathways. Having understood that, we also figure out over time of the last half a century or so, that many of the diseases which are genetic especially are due to the fact that there are some problems or inadequacies or problems or deficiencies in receptor-ligand binding. So, for example, if you genetically inherit a disease from your parents or your grandparents, it could be that it has to do with the fact that you have inherited a gene which somehow impedes the kinetics of receptor-ligand binding related to that biochemical pathway. As a result, that disease is manifest in you. You see what I am saying? So, it is not necessarily that all diseases would be manifest because of a certain biochemical pathway, but for example, diseases like high cholesterol or diabetes or things like that, and many of these diseases has receptor-ligand binding as a part of it. So, we will look at receptor-ligand binding as a phenomena first, as a mechanism first, and then we will look at one particular disease, which is familial hypercholesterolemia. That is again a genetic disease, and we will try and understand that what are the factors that lead to this genetic disease, and it turns out that we will figure out that much of it has to do with improper receptor-ligand binding, receptor-ligand binding that should have been that isn't the way it should have been. So, having said that, so let us start the chapter essentially. So, it is called receptor-ligand binding, and it is 6-7 lectures we will have on this, probably the longest of the chapters that we will do. So, what are receptors? Before we try and understand receptor-ligand binding, what are receptors and what are ligands? Receptors are proteins that are present in the cell membrane and the organelles, including the nucleus, and I will show you pictures in maybe less than a minute of how the receptors look. So, these are proteins which are present on the cell membrane and that of the organelles, and what is the role of receptor-ligand binding, or receptors, so to say, is to transfer cell signal from the cell interior to the cell exterior. So, these receptors are in a way connects between the interior of the cells to the exterior of the cell. Okay. Now, one of the things that I keep telling probably not in this lecture, but in other classes, is that there is something called an inherent wisdom of the cell. Okay. The cell itself has a self-protecting mechanism, just like the body itself has a self-protecting mechanism or self-defense system, either a self-defense system or a self-protecting mechanism. So, you are in an environment, for example, where there is low oxygen, and you would be instinctively you would be guided by your body to move away from that environment to an area where there is higher oxygen, or if there is a fire out there. Instinctively, even if you are if you are not thinking, your body would instinctively take you away at that. So, there is a self-protecting self-defense mechanism of the body. Similarly, each cell is a full-fledged, almost like a full-fledged individual, because come to think of it, the cell is a building block of the body, and the whole of life on earth itself started with a single cell, not with with multiple cells. So, come to think of it, we are just a conglomerate of cells, right. So, and there is each cell has a wisdom, okay, is wisdom of its own, which is to protect itself. Number one, and the first priority is to protect itself, and B is to perform the function that it is supposed to perform. Okay, when it cannot perform the function that it is supposed to function, then it dies. And in this process called necrosis, I think I have spoken about that sometime earlier. So, so the process, what I am trying to talk about is that so the cell as a whole has this mechanism of self defense and self protection, okay, and for that it needs to function on its own. But again, the cell, one particular cell, needs to act as a part of an organization, okay. That organization, you can call it a tissue, an organization of tissues is called organs, and an organization of organs is called the human body. So the cell has to function from the level of a single cell to the level of the tissues to the level of the organs to the level of the human body, right. So how does it communicate with the exterior? It is not enough to, for us, for a body, for example, you want to preserve your body and preserve your system, right. At the same time, you need to communicate with the external world, you need to communicate with your friends, with your teachers, with your family, for different things, because at the simultaneously we are a self preserving system, at the same time we are part of a larger world. So there is a necessity to communicate and there is a necessity to preserve oneself. The reason to communicate is sometimes, for example, the hydrogen ion concentration inside the cell has grown to a level, say, higher than, such that pH is higher than 7 is lower than 7.2, it's become more acidic than it's supposed to be, something like that, right. So then the cell needs to communicate to the external environment to kind of balance this, right. The cell needs to communicate to the external environment to kind of balance this, so that the pH is decreased within the cell, because acidic pH is not good for the cell. So these kind of communications are not just necessary to work as a group, okay. For the cells, it is not just necessary to work as a group, but it is also necessary for the cells to preserve itself, and the receptor ligand binding or receptors themselves act as a very important source of that, okay. So these are the sort of quote unquote the neurological, not really, but the neurological connects between the cell and the outside environment, okay. So they are the ones who allow the cell to communicate with the outside environment, and these, if I go to the screen now, so these are receptors are essentially proteins that are on the cell membrane. It could be on the organelles also. For example, why on the organelles? Because if the nucleus wants to connect or communicate with the cytoplasm, then the receptor in that case is going to be on the surface of the nucleus, sticking out of the surface of the nucleus inside the cytoplasm. Otherwise, if the cell is trying to connect with the outside environment, then the receptor is going to stick out from the cell membrane into the outside environment. I will show you a picture how it is on both sides of the membrane, and the major job of this receptors is to transfer signal between the cell surface and the cell interior, or between the different parts of the cell. If it is the case, if it is between the nucleus and the cytoplasm, then between the different parts of the cell. If it is across cell membrane, then it is transfer signal between the cell surface and the outside. And what are these receptors? These receptors are G protein type receptors, ion channel receptors, enzyme linked receptors, receptors with intrinsic enzyme activity. You have read some of these, I think the protein receptor, the ion channel receptor, we had discussed this in a previous course. But so these are so all of these means are essentially to communicate between the external and the internal environment. For example, the protein channel or the ion channel receptor, for example, if you want to throw away a certain ion, say sodium or say potassium or say hydrogen, then this receptor would give that signal to the system saying that the ion channels have to be opened out and you want to throw out this particular ion. So that is the function of these receptors. Now what about ligands? So ligand could be a peptide, a protein or a hormone that binds specifically to the receptor and helps in transfer of information. So it is like a mechanism where the ligand, so the receptor just sticking out into the cytoplasm does not make much sense. It has to bind with the ligand and transfer information. But look at two words which are in bold over here, transform information, and the other one is specifically, okay. So that is important. So which means that it is like a lock and key mechanism. So a specific receptor will bind to a specific ligand, okay, that is the whole idea. So there is a lock and key mechanism, just like in the case of enzymes, enzymes will bind to a particular substrate. So similar kind of thing. And now what the idea of the receptor ligand binding. So we talked about the receptor, we talked about the ligand, let us talk about the binding that follows. So how does this binding occur? Similar, very similar to the enzyme binding to a substrate. So what happens is when a ligand binds to the receptor, and I will show you a picture in a minute, it produces a conformational change. This I won't be able to show, but you can sort of imagine it. So it produces the conformational change in the cytoplasmic portion of the receptor, which in turn initiates a cascade of signal effects that results in functional changes in the cell. So, whenever these, so you want to transfer a signal, that is your job, right, that is the reason you are doing this binding. Now, you have a set of ligands which will bind specifically to the receptor. Now, as soon as the receptor binds, there is a to the ligand, there is a conformational change that happens in the cytoplasmic part, and I will show you these two parts, and the cell signaling starts, the process is initiated. Okay. And then after this process is initiated, small molecules and small molecules which are secondary messengers, they may be activated and enzymes may modify the protein also. I mean, this is something that happens after the conformational change occurs, after the cell signaling process starts. You can have secondary messengers, you know, in pulmonary system you have certain kind of messengers, T cells, and you know, different kinds of secondary messengers which are activated, and you can start the process which are related to secondary information passage, not the primary one. So, if you look at this picture over here, so look at this. So, you have over here what we have is a set of ligands which are, these are labeled ligands, okay, and these are general ligand. So, what is the difference between a label ligand and general ligand? The difference is that I have just taken here I know that this receptor is going to bind to a particular kind of ligand, and I label that ligand, and I mix it up with other ligands, or other ligands are also there automatically, you know, generally they are there in the system. So, I have no control over this. So, the label ligands are the ones which are specific to the particular receptor. Now, you see what is happening over here, look at this picture and look at this picture. So, at the end of the binding process, you see that the label ligands only and the label ligands only have bound to the receptor. The reason is specificity, because they are the ones that are allowed. Also notice this receptor over here, this is how receptors are always drawn, by the way. You know, when you draw a receptor on your copy, this is how you draw a cup, half a cup shape over here, and a spring like structure at the and tail. So, and the tail is in the cytoplasm, and the cup like structure is ejecting out of the cell membrane. Okay. And the cup like structure is such that, the reason the cup like structure, as I said, is a lock and key mechanism. So, this receptor ligand can come and sit over there, fine. Now, look at this, what is happening, this is a nucleus, the blue one out here. Look at what is happening in the cytoplasmic part of the cell from here to here. There has been a conformational change which is denoted by this. Okay. So, that is the thing. So, ligand population which consists of the labeled ligands and unlabeled molecules, in order to quantify the amount of ligand that binds, and binding produces a conformational change to the cytoplasmic portion. Okay. So, the next thing that we want to do is try and quantify this in terms of the kinetics. So, what do you think could be the kinetics of binding? No, it is not going to be Michaelis-Menten, but similar to the enzymatic binding. It is not going to be Michaelis-Menten, the reason being, you know, the steps of the pseudo steady state equation and all those steps are not exactly the same, but the equation in general looks like that. It's the kinetics actually a lot simpler. So, let us talk about the kinetics. So, ligand is denoted by L always, R receptor, and C is the complex that is formed. Okay. So, K1 is the association rate constant, which is the second order rate constant, as you can see, molar inverse times minute inverse, or molar inverse times second inverse, and K minus 1 is a dissociation rate constant, which is minute inverse or second inverse. Okay. Is it right? Go on. Okay. So, these are examples of cell surface receptors that are there in the human body, and you know some of these. For example, let us go through them. Epinephrine. Epinephrine is a well known neurotransmitter, it is a pretty well known neurotransmitter, and it regulates metabolic activity, you know, it regulates hormones and that kind of nurture metabolic activities, and it is a, as I told that there are different kinds of receptors, and as you can see in the middle column, it is a G protein coupled receptor. Okay. Serotonin, the next one is serotonin. Okay. Serotonin is a neurotransmitter that causes a constriction of blood vessels. It is very well known. Have you heard of serotonin? dopamine, you have not heard of serotonin and dopamine. These are very well known neurotransmitters. For example, if if you have a friend who is depressed or something, what do you give, what is something that you typically give to that friend? Something to eat? What kind of things to eat? Yes? No idea? No idea what do you give to a friend who is depressed? Chocolates, right? Typically that's what is given, at least in the, so why is that? Because chocolates has these chemicals which, if you look here on the screen, which stimulates the production of serotonin and dopamine. Okay, and these are, so if at any point of time, if you're feeling sad, just go buy, eat some chocolates. So that helps. It's in terms of cholesterol or whatever, may not be very good, but it is well known that chocolates has a lot of neurotransmitters in there, has chemicals that stimulate these neurotransmitters, rather, serotonin and dopamine, and as a result these are produced more in your brain and goes into the bloodstream, and constriction of blood vessels in the brain. As a result, you feel this this state of happiness or joy or whatever. So this whole, every emotion that you feel is essentially a chemical reaction. Come to think of it, quite sadly so. So your whole expression, when you feel good about something, when you feel happy about something, essentially being just the fact that serotonin and dopamine are being produced in the right quantities. Now, if you do not have them in the right quantities, you feel depressed or sad or low. If you have too much of them, you feel euphoric. So these are the chemicals, these I'm not sure if you had been taught in the other part, but these are some of the chemicals of life, the serotonin and dopamine. And all these actually are chemicals of life. So similarly, acetylcholine is an ion channel receptor, and it also is a neurotransmitter. So these three are the major chemicals that work, neurotransmitters that work in the brain. So serotonin, dopamine is not listed here, and acetylcholine. So any sort of feeling of goodness that you have at all points of time are a permutation and combination, or a total production of these three things. It's not God's gift to you. Happiness is not God's gift to you. It is just these three chemicals, serotonin, dopamine and acetylcholine, working together to produce that emotion. Similarly, other kinds of emotion are produced. As I said, sadness is produced by by a depletion of these chemicals. Okay. Now, next, so these kind of form the same group. Epinephrine is one that stimulates hormone activity, and this is also important. Epinephrine also kind of takes part in this. So this is kind of one block you can think of it. This is a G protein. This is a G protein. This is ion channel. But these are one block of neurotransmitters that govern a lot of emotions and a lot of feelings in the brain that you have. Cytokines is a different kind of receptor. Okay, it's not this neurotransmitter group. Cytokines, I'm not sure if I've talked about this, but cytokines are essentially immune cells. Okay, so monocytes and macrophages, these all belong to cytokines. And did I discuss the effect of cytokines and things like that? Maybe I will. Did I discuss before the effect of cytokines, how I think macrophages and stuff like that. I think we'll do this in this course also, where I can give you a little briefing, quick little briefing. I probably have a picture out here. I can show you the picture, and yeah, I think so. This is receptor mediated endocytosis, but similar is a process for just let me see, I might have a better picture. Okay, so this is probably the best picture I have. So cytokines don't really work the exact way, but this endocytosis process is the same. So if you do not think of this as a cell in general, but if you do not think of this as a cell, if you think of this as a cytokine, then this is exactly what happens. So the cytokine will form these parts. So cytokine is like, say, a macrophage. Okay, will form these parts which are known as invagination. Okay, so a part of the cytokine deforms itself, and say you have a So, deforms itself like this, like a mouth shape, and I will go into the details later, and forms this kind of thing. You can see here this kind of structure out here. So, why does it form this kind of structure? Because the bacteria, so these are immune cells, and so where the bacterial attack, let us talk about. So, what happens when you have a bacterial attack? For example, you inhale, you breathed in certain bacteria, or you drank something which had some bacteria or something like that. Let us talk about the simplest case where you inhale, somebody sneezed in front of you and you inhale certain bacteria. Now, if your immune system is good, then you are not necessarily going to fall sick. If your immune system is not good at that point of time, it is a function of time also, then you might fall sick, right. So, what happens when the bacteria enters your body? What happens is that it goes through the larynx, you breathed it in, it goes through the larynx, pharynx, trachea and so on, and goes into the lung. Okay. Now, in the lung, what you have is that you have the cells on the lung, and on these cells you have around 7 macrophages which are there on these cells. Now, per cell. So, it is a huge, and on the alveolus actually. So, each alveolus actually will have 7 macrophages. So, it is like around a couple of billion macrophages, I think, in the lung itself. So, each macrophage, if I now can go to the screen, so each macrophage is like a cell like this. Okay. So, what happens, the bacteria comes and says the bacteria has entered your alveolus. Alveolus is a little sac like spaces inside your lung, entered it, and it is out here, somewhere out here. So, then the process starts. The process is, first step in the process is that the macrophages, which is small in size typically, increase in volume a lot. Okay. 4 times, 5 times increase in volume. First thing is the increase in volume. As a result, the energy requirement goes up. So, it changes, the macrophages changes state from what is known as P state, P state to war state. Okay. So, the body, so the process starts with again a receptor ligand binding. So, there is something called T cells, when there is some receptor ligand binding that goes on, and they sense these bacteria that are there in the system. Okay. So, once they have sensed these bacteria that are there in the system, they send some signals to the central nervous system saying that this process, there is a bacterial attack in the system. Now, once the bacterial attack has been sensed by the brain, by the central nervous system, then it sends out signals to the, so all these signals work through neurotransmitters and essentially receptor ligand binding. So, that is the whole job of receptor ligand binding. So, these signals are sent back to the lung saying that there has been an attack. So, it is like an attack on a country, for example, and the country goes from the peace state to the war state. So, what does it do? It deploys soldiers to the boundaries of the nation, right. So, similarly, the brain tells the lung that there has been an attack on the system, a bacterial attack on the system, or a viral attack. It could be a viral attack, it does not have to be bacterial attack on the system, and we need to deploy soldiers. So, what do you mean by soldiers? The soldiers here are the immune cells, the cytokines kinds. In this case, in the lung, so different kind of cytokines you have in different parts of the body. So, the cytokine kind that is important in the lung is called macrophages. In the liver or the kidney also, there also you have macrophages on the guts, and you have other kinds of cytokines as well, as I said, monocytes and so on in the blood. So, the cytokines are alerted. Now, once the cytokines are alerted, then what happens? They say that the brain tells the cytokines that increase the volume, because unless they are big in volume, so they cannot beat the bacteria or the virus. Now, when they increase the volume, we need a lot of energy, okay, to increase, not just to increase the volume, but to sustain it. Why? Because an increased volume, just as we read in the cell growth process, an increased volume will necessitate increased metabolic activity. So, increased metabolic activity means increased use of energy. And have you ever noticed that if you have a bacterial attack, even before you have a fever or anything, you start to feel very weak, right? Or a viral attack, in either of these two attacks, even before you got a fever, you start to feel very weak. Why do you feel weak? Because your energy is now being diverted to your immune system. Okay. So, your immune system is being pumped up while the rest of the body kind of takes a setback. So, the doctor tells that eat well, if you have fever, that eat well, eat enough glucose or stuff like that, drink enough water, especially water that has carbohydrates in it, or these kind of is saturated with glucose or some kind of carbohydrates or other. So, so you do that, so that you provide metabolism to the cytokines. Now, the cytokines then expand in volume, Then let us now go back to the state. So, once it expands in volume, you form these invaginations. Why does it form these invaginations? Because it wants to engulf the bacteria. Okay. So, the bacteria, so around the place where the bacteria is there, it forms these things, and if you have 7, for example, one alveolus will have 7 macrophages, so 7 these macrophages will form invaginations, and one macrophage can form multiple invaginations. Okay. It forms these and it waits for the bacteria. It is like, you know, trying putting a fly on and waiting for the fish. Okay. So, it waits for the bacteria or the virus to come here, and as soon as it comes here, it entraps it. It entraps it like this, and then it forms, and then it joins its both, you know, both. It is like a mouth of the macrophage. It kind of eats the bacteria up, closes its mouth, and this is what is formed. You see here, this where the arrow is. This is what is formed out there. Okay. So, and the bacteria is entrapped now here. Now what? What do we do? You have a living bacteria inside a macrophage. The macrophage cannot afford to keep that bacteria just like that out there for a long time, because the bacteria might start to multiply and infect the macrophage itself. So, what would it do? What do you think it should do? Yes, yes. So, that is what it is going to do. It is going to acidify. Why is it going to acidify? Because essentially what it needs, what do you know, when you want to kill bacteria in the drain or anywhere, what do? Use either acid or phenyl or something like that. Essentially at the end of the day, what you use is a bleaching agent, okay, which is what, either chlorine or hydrogen peroxide, a bleaching agent. So, the cell, the macrophage also uses a bleaching agent. The bleaching agent it uses is hydrogen peroxide actually, and hydrogen peroxide is not there in the system. So, what it does, it produces hydrogen ion, okay, and the hydrogen ion then reacts with the water to form hydrogen peroxide. Okay. So, what happens here is that this is known as the vesicle once it is kind of taken in, and then it acidifies out here, which is that hydrogen, it pumps in hydrogen ion. Where does it get the hydrogen ion? The hydrogen ion is present inside the cell. Okay. So, it keeps pumping in hydrogen ion, and then there is, you can say that yes, there is going to be a depletion of hydrogen ion. Yes, there is going to be, then these other ion channels are going to open up and hydrogen ion is going to come in from outside, and then it is going to pump in hydrogen ion inside these vesicles. It pumps in hydrogen ion inside these vesicles, it becomes acidic and the hydrogen peroxide is formed, and that kills the bacteria. Now, once it kills the bacteria, it does not want to keep dead bacteria inside itself, it throws it out, okay, and then this invagination is gone and it is ready for the next to engulf the next bacteria. So, this is the process, you know, I kind of digressed, and I am supposed to teach this anyway a little later in the course. So, kind of digressed and went forward. So, this is the cytokines stimulate immune cell activity, and this is how, you know, how the immune cell activity works. Then there is interferon. This is again, so these are neurotransmitters, the first two, and the next two are ones that stimulates immune activity. So, this is interferon is also a cytokine, but it is a one that interferes with the replication of viruses. It is different, you know, it prevents the viruses from replicating in your system. Then you have insulin, you know, you are all aware of insulin. It increases glucose uptake in the cells and functions as a growth factor, you are aware of that. And then other growth factors, receptors that have intrinsic tyrosine kinase activity, which stimulates cell division. You know, we studied this, the whole cell division and the cell growth process earlier, and there are these cell growth factors which we did not study at that point of time, which are essentially receptor ligand binding, which stimulates the growth process, okay, by enhancing metabolism and by different other ways. So, what I am trying to give you is a sort of a landscape picture of the different types of cell surface receptors that are there. There is one block of receptors which are neurotransmitters, extremely important for the body. There is another block of receptor which are immune which have immune activity, which are equally important for the body, and the third kind of receptor is the one that stimulates growth activity. So, they are all equally important. Just as you need to grow, you need to protect yourself, you need to feel happy also. So, you know, all these three are important. So, let us now go to the kinetics of receptor ligand binding, and as we said that the receptor ligand binding is a process of given by the second order reaction, forward reaction, and the first order backward reaction, a reversible system of L plus R giving C, and K1 being K and K minus 1 being the forward and the backward rate constants respectively. So, if you write the equation, it is simply del C and del Cc del T, Cc here being the concentration of the complex, times K1 Cr Cl minus K minus 1 Cc, right. Now, what we're going to do is, in this system, whenever we are doing most of this analysis, we are not going to measure things in terms of concentration, but in terms of numbers, because these receptors are, isolated points essentially. It is not a continuous system. The receptors are, you understand what I am trying to say. So, if you are measuring a concentration in a liquid, it is a continuous system, whereas receptors are isolated points on the cell, and we can track the numbers of these receptors by labeling them and looking at them under a microscope. So, we are not going to measure their concentration, but we are going to measure their numbers. So, if I go back to the screen, so you see here that NR is the number of receptors per cell. These are the terminology that we are going to use, and you may note it down because this is something that we are going to use consistently. So, NR is the number of receptors per cell, free receptors. NC is the number of complexes per cell, and nRT is the number of receptors per cell which is either free or bound. So, either it is in the complex form or in the free receptors form. So, nRT is essentially equals NC plus NR. Okay. So, this is written, it is valid under limited conditions. This equation is valid under limited conditions. Can any of you sort of intuit why you think it is valid under limited conditions? Some of the receptors are ejected.
Inhibited by inhibitors like other ligand.
Yeah, there are several things where one of this is correct what you said, that the other ligands can bind to the receptors. The receptors might die. Those are more important things actually, because even if other ligands are binding, I can add in NC1 plus NC2 plus NR, still there is a way to write a balance. But the major problem is receptors are not, they do not live forever. So, they die, there is a recycling that is going on, and they are again created, they are born also. So, those are things that have to be taken into account when we actually do this. So, but under limited conditions, for simple cases we will first assume this is true, and then we will look at cases where this is not true also. Okay. Now, what about the ligand? Now, the ligand typically is in the concentration form. The reason the ligand is in the concentration form is because the ligand is in liquid form. Okay. So, ligand is measured in terms of concentration, whereas the receptor is measured in terms of number. So, there has to be some conversion that we have to keep doing all the time. So, in experiments, ligand is added in solution in a ligand form at an initial concentration of Cl0, and if the ligand, so again you can write some sort of a balance equation or some sort of a constraint equation for the ligand, that if the ligand is not metabolized by the cell, then either it is in solution or bound to receptors. Okay. Just like the receptor is either in the free form or in the bound form, similarly the ligand is either in the solution form or in the bound to receptor form. Okay. So, if n equals the number of cells per unit volume and Na is the Avogadro number, then Nc is the number of, just as we define, the number of complexes formed times n over Na, that will give me the concentration of the ligand in the bound form, correct? Okay. And Cl is the concentration of the ligand in the free form. So, if you start with the initial concentration of Cl0, and we are assuming that ligands are not being added at any point of time after the initial time, then Cl0 plus Cl0 equals Cl plus n over Na times Nc, fine. So, now what I want to do, I had my equation over here, right. So, I want to replace this in one particular way. So, it is in terms of n, say. So, let us replace this here, Cr and Cl, everything in terms of n. Then what you get is Cc, it turns out the concentration of the complex is n over Na times Nc, is it correct? Because just multiply by the number of cells per unit volume divided by Avogadro number, you will get concentration per unit, moles per unit volume. So, concentration of complex, and Cr would be n over Na times Nr, fine. So, what we do, we go back here and replace these concentrations by this. So, K1 n, this will be nRT minus NC, y nRT minus NC from this, okay. And this will be here from here CL naught minus Na over Nc, CL would be CL naught minus Na over Nc minus K minus 1 Nc, right. So, equation 4 what it gives us is, now I have been able to convert my entire concentration equation into a number equation, fine. So, now the next assumption that comes in is that Na over Nc is much less than Cl naught. The reason being that you always provide ligands in excess. When you do the experiments, your ligands are not the limiting reactants, it is typically the receptors that are limiting reactant, and that is also physiologically true, because what has to bind typically is in excess. Typically you can always have always have occasions or times when the ligand is not in excess, but typically for example, especially in pathophysiological conditions when there are diseases, the ligand may not be in excess, but typically it is a ligand that is in excess and the receptor is not in excess. A ligand that is in excess and the receptor is not in excess, right. So, if we can do that, then we can simplify this from equation 4 here, okay. This part we can ignore and we can simplify it a little bit, and so nRT, this also we can ignore, and sorry, and NC you can take it into this. So, sorry, and this NC you can take into here, and this part, then this part over here you can ignore, right. If you look here, why one of the reasons mathematically it helps us to do is solve the equation itself, okay. Because if you look here, it is I think it still could be solved, and you can try solving it, because you still use partial fractions and solve this. Yeah, to my mind you can still solve this using partial fractions. You just break open the bracket and regroup them as separate fractions, and you can solve them. But here, for example, it is much easier, and you can do an exponent. This is a more or less a valid assumption. You can do an exponential you can get an exponential solution out of it, okay, straight away. So, if I write my equilibrium constant as k minus 1 over k1, then this can be slightly simplified as this, okay. Now, can you look at the equation 8 over here on the screen, and just tell me how it's going to look like if I plot my NC, the number of complexes that's formed with time. So, NC naught, okay, so NC naught is the initial number of complexes present in the system. Let us, for the sake of simplicity, assume that it is 0, you didn't start with any complex at all. So, how is it going to look now? Yeah, I mean that is as simple as that. If you do not have the first part, if you have the first part, then there is an added time part to that, and otherwise it is just saturating out with time. It is exponentially growing and saturating out with time, because the t going to very large time, this has to saturate to this value nRT Cl naught over Kd plus Cl naught, fine. And if you have the initial part, that is NC naught is not 0, then you have another exponent out there. So, you can draw the two exponents and then add them up with a summation of the two exponents, okay. I will show you the picture in a minute. So, from this we come up with the concept of half time, okay, the concept of half time, and this is just like half time in radiological decay, and for the way it is defined is that for a given ratio of NC over nRT, time required to reach half its maximum value. So, the maximum value for the complex that is going to be formed, half of that, when is half the maximum value attained, okay, that is my half time. So, NC max out here you can see, if if NC naught is 0, let us assume for the case of simplicity, then NC max is simply this, right. So, whatever at time going to infinity, if this is not 0, then it will be NC naught plus this, okay. So, your NC max. So, all you need to do is half of this. So, NC over NC max equals half, okay. So, NC max is well known over here, should equal this, Is it clear why? Because this is NC max. I can write at, see, if you ignore this part, if NC naught is 0, you can ignore this part, and NC could be written as NC max, which is this thing over here, times this. So, for half time you just need to write NC over NC max equals 1 minus exponential this thing in the parenthesis curly brackets equals 0.5, clear. So, this is what we do, and you can solve for your half time, and you get comes out similar to what you get in logical decays. So, half time is ln 2 over k minus 1 times 1 over k L naught plus k d. Okay. Now, for small ligand concentration, if CL naught is small, then T half equals ln 2 over k minus 1. Does it make sense? You are all nodding, but does it make sense? It does. Let me go to the screen and show you this. First thing, first one for small, huh? Not zeroth order. For small ligand concentration, T half is given as ln 2 over k minus 1. Is that, does that make sense? Yes. No, it doesn't make sense. It's oxymoronic. What would we start with? Is?
Not limiting.
It's not limiting. It's very high. And then we cannot assume that CL naught is very small, because as soon as you make it very small, it also becomes small as compared to NC, the concentration of the complex, right. So, this is, though it's theoretically it makes sense, but it's oxymoronic in general, because you cannot assume it to be first a very large concentration and then make assumptions and simplifications based on that fact is very small. The second one, of course, makes sense, that if CL naught is very large and KD is very small, then this term is very high, and then T half will go to 0, which means that within a very short period of time the half time would be reached, and the NC max, half of NC max would be reached within a very short period of time. But that, remember, has to do with not just scale not very large, but also KD very small. Okay. KD being the, what is KD? KD is the dissociation rate constant. So, here the backward over the forward rate constant. Okay. So, KD being very small means that the forward rate constant is much higher than the backward rate constant. Okay. Which means that not just that the ligand is present in a large amount, but the reaction is taking place in the forward direction very fast. So, these combined effects of these two facts, that the ligand is present in large amount and the reaction is taking place in the forward direction much faster than in the backward direction, will make my half time very small. It is physically meaningful. Okay. Now, let me show you this plot. So, this is how it looks like if I had if my NC, initial NC is 0, this is what it is, NC naught equals 0, then this is how it looks like. So, as you see, CL, it depends on CL naught over KD, and these are various values. So, it saturates out to a value. If I look at, it will saturate out to this value. Okay. And this one NC max, I can write that as nRT times NC max, I can equals nRT CL naught over KD plus CL naught, and if I divide all through by say CL naught, I get CL naught over KD plus 1 plus CL naught over KD. So, that is what we are doing in the plot over here. So, CL naught over KD is a single parameter. So, I have been able to convert this two parameter into a single parameter system now, and as it changes, it increases. Now, this what does this remind you of now? Yeah, this reminds us of the Michaelis-Menten kinetics. This is exactly the way I wrote it over here. If you look here, so CL naught, this is exactly looks like the Michaelis-Menten kinetics, right? So, this is it, and let's So, I want you to, why don't you write down this problem? I want you to write down this problem, and then I will give you another problem. What I want you to do is that do these problems on a sheet of paper and submit it in the next class next week as an assignment with your name and roll number written. This, and there will be another one I will give at the end of the class. So, determine the half life for binding of endothelin to endothelin 1 receptor under the following conditions. Determine the half time for binding of endothelin to endothelin 1 receptor under the following conditions. K minus 1 equals 0.005 minute inverse, Kd equals 16 picomolar, Cl naught equals 110. So, these are different values, you know. So, I want you to do for these different values 110 and 100 picomolar, and 1 picomolar is defined as 1 into 10 to the minus 12 molar. Finished? Okay. Let us go forward. Now, what the next thing that we want to do is the determination of the rate constants of the receptor ligand binding. You want something? I will repeat one more time. Determine the half time for binding of endothelin to endothelin 1 receptor under the following conditions. K minus 1 equals 0.005 minute inverse, Kd is 16 picomolar, Cl naught is 1, 10 and 100 picomolar for 3 different values, 1 picomolar being 1 into 10 to the minus 12 molar. Okay. So, the next thing that we need to do is some of these constants we need to evaluate, because otherwise there is no way forward. How do we evaluate these constants, and then later on during the course we will try and understand how we evaluate these constants experimentally, not just theoretically. So, right now let us talk about how to do some plots and evaluate these constants. So, my expression, the kinetic equation is given like this. So, in this is a case where NC naught equals 0. So, it is given as nRT Cl naught over Kd plus Cl naught times 1 minus exponential this factor out here. Now, the kinetic parameters here are the forward and the backward rate constants K1 and K minus 1, because you know you have the dissociation constant and the backward rate constant coming out separately. So, you have to, there are two constants, there is no running away from that. nRT being the total number of receptors, that is an unknown, and we need to perform experiments at various values of the. So, one of the ways I will I would not talk about the experimental procedure, I will talk about that later in the course, but right now theoretically if you look at it, you know, without thinking of the experimental procedure, theoretically one of the ways is to perform these experiments just like as you do. This is like a first order system, and you know, as you do in all kinds of why is this a first order system, by the way? Is this a first order system to start with? Is this a first order system or not? Yes, it is. Why is it a first order system? Equation is same. Why? Because of the assumption. Why is that? Because of the assumption we made that the ligand is present in large amount. Otherwise, it is a second order in the forward direction. So, when we study the kinetics of the system, it is simply as simple as the kinetic study of that we did for a first order system. So, you perform experiments at various values of the Cl naught, the ligand concentration, and then as I told you, as I showed you just before at the beginning, is that essentially how do you do that is you tag the ligands, okay, the ligands that you want to. If they are not tagged, you cannot measure, you tag them with a fluorescence, you know, make them fluorescent so that they will glow, and that is the way you measure them. Okay. So, this is the formula, and if you do not think about all the details of the process which we will deal with later, you essentially are carrying out your experiment. If you look at your problem, last problem, what we did was Cl naught, we carried out different values of Cl naught, we carried out the whole experiment at different values of Cl naught. So, here also you carry out the experiment at different values of Cl naught, and you measure the amount of constants and amount of complexes that is formed, because these are all tagged. So, these are fluorescent. So, because they are tagged, you can measure them in the tag form, right. So, you measure NC as a function of time for different values of Cl naught. You start, you know how much Cl naught you are putting in, and you measure your NC because it is tagged, and you know, so it will emit fluorescence. So, you measure them with time as a function of time for different values of Cl naught, and as I showed you last time also just here, that Cl naught over Kd is a parameter, you know, you can divide the whole equation by Kd. So, Cl naught over Kd would be a running parameter, and as you keep increasing your Cl naught over Kd, the slope would be changing, right, because as you keep increasing your CL naught over Kd, what does it mean? That the that the reaction should proceed in the forward direction more, because you have increased the amount of ligand that is present and you have increased the forward rate constants, right. So, I will just go little quickly because I need to want to go to the problem. So this is so the slope here, let's be a little quick. So, if you take the log of this, so 1 minus NC over NC max on this side, log of this here, okay. So, this is my NC max, by the way. So, NC over NC max you can take, so NC minus NC over NC max is 1 minus exponential of this term, right, which means that 1 minus NC over NC max is simply the exponential. Now, you can take log of both sides. So, you will get ln 1 minus NC over NC max equals minus K1 1 plus CL naught over Kd. Is that correct, Lisa? Times T. Okay. Now, at steady state, NC equals NC max. Steady state means, what does steady state mean? Infinite time, yeah, t going to infinity in this case. This is the only steady state possible, no other steady state. Okay. So, the slope here is simply this. If I plot this 1 minus NC over NC max, I know how much my NC max is going to be, because I know how much ligand I gave in, and so if I plot this 1 minus NC over NC max, so I once it attains steady state, I can measure my NC max. If I plot this with time, then this is my slope, and from the slope so CL naught is known, right, and for different values of slope I can get these constants, okay, because so with CL naught, okay. Now, so this is one. So, you measure this slope for different values of CL naught. Next step is this one, the plots the slope. Once you calculated the slope for different values of CL naught, plots the slope for different values of CL naught, okay. And then from this you can calculate the intercept, from this the intercept and the slope of this, you can calculate both the constants. Is it clear? Okay. So, there are two unknowns out here in the slope, but what you do is you now again plot the slope with CL naught, and from the intercept and the slope of this. So, the intercept will give you just k minus 1, I believe, and the slope will give you k 1 or something like that. Yeah. So, yeah, slope will give you k 1 and the intercept will directly give you k minus 1. So, you get both the constants. So, this is one approach, and the other approach, very quickly let me do, maybe, and if I need to I will come back next class. So, to obtain nRT, use NC max equals nRT times CL naught Kd plus CL naught. Now, NC max is something that you know, right, and then you can back calculate and find your nRT. And this is the plot that you do, and I will come back to this later on. The plot that you do to do this, so essentially you want to get your nRT, and this will be a intercept of this, is known as Scatcherd plot. This is a very important plot, and I will come back to it again and again. This is known as Scatcherd plot, okay. NC over CL naught, you might want to make a quick note, or at least of the axis. So, the way you do it is you make a Scatcherd plot of NC over CL naught, and that is receptors per cell, cell inverse, that is the unit of this, and over NC. And Scatcherd plot is what kind of plot? Is it a steady state, unsteady state, what kind of plot? What kind of plot is Scatcherd plot? Steady state plot, right. So, essentially this is what you are plotting, this box out there, okay. So, NC max over CL naught, though it is written, I have written here NC, but it is actually NC max that you are plotting, and NC max means that steady state has been attained, okay. So, NC max over CL naught equals nRT over Kd minus NC max over Kd, fine. So, if you plot this over this, then what do you get? You get your intercept is this and slope is 1 over Kd or minus 1 over Kd. So, you can directly calculate your nRT. So, in a way, as I said, that if you know your Kd, if you like, you already know your CL naught, if you know your forward and backward rate constant, you can know your Kd. So, you can back calculate from NC max to calculate your nRT, and this is the process. The reason we assume this process is because we do not need one data, we want to use multiple data points to minimize error. So, this is the way. So, Scatcherd plot, remember, most important thing to remember, it is a steady state plot. What you plot essentially is the steady state value of NC over CL naught versus CL naught. The intercept will give you nRT over Kd, the slope will give you minus 1 over Kd. Okay. So, the last thing I want you to write is this problem. So, these two problems you are going to do together and submit as an assignment in the next class. So, the problem is use the following data to determine the specific binding of a ligand to a receptor. The values of Kd and the number of receptors per cell. Okay. So, basically you need to find all the rate constants and nRT, and CL naught is given in molar 110 to the minus 10, 510 to the minus 10, 110 to the minus 9, 510 to the minus 9 and 110 to the minus 8. So, first case is when there are no unlabeled ligands there in the system, and second case is when there are 100 excess unlabeled ligand, and that is the data that is given to you. So, 100 excess unlabeled ligands per cell, by the way. Okay. And these are NC values, that thousands are numbers that are given in thousands are the NC values you obtain. NC values are steady state, or in other words NC max. So, the hint is obviously you have to use something like a scattered plot to do this, because this is steady state values that are given, and but there are some tricks involved in it, and some just okay. I hope you have all written down. So, we can stop here and we will continue in the next class. Thanks.